Okay, so today we're going to talk about how to generate a Geneious variant call report file. And this is a question that we get frequently from clients who are using, some of our clients are using Geneious, and they want to know how we generate our standard variant report file. And we're going to show you how to do that today. And we're going to assume that you already have Geneious, you've got Geneious Prime launched and running, that you've already imported your sample FASTQ files. You've already run the sequence alignment variant call algorithms against those FASTQ files, those sample files, and now you want to know how to generate the variant report. So that's where we're going to start. So anyway, in this case, we've already got Geneious launched. If you look, there should be a config file to have these three sort of diagonal bars. It'll say something like, in this case, it says demo bacteria Geneious. You'll have your sample name in here. And it'll say that is assembled to some reference genome, and in this case it's Staph aureus. Okay, so it'll be sample name assembled to reference genome. The first thing you want to do is check that record. Oh, and by the way, this is the document field or document panel in Geneious up here. That will bring up three other panels, or at least it should. And this may depend on where you're at with Geneious. If you've already run Geneious a few times, it may look a little bit different than this. It depends on how you save things and configure things. But you should be able to bring up this sequence view panel in the middle. And on the right, there will be this configuration panel. And then down on the bottom, there will be a sort of a spreadsheet panel. The first thing you want to do is make sure that you look at this tab list along the top here and click on config view. You want to make sure that that's highlighted. The next step is to go over to the configuration panel on the right, and then you'll see this list of options along the right panel. The one you want to choose is this yellow right arrow. Click the annotation and tracks panel, and that'll bring up this view. And then what you want to do is make sure that show annotations is checked. So I would do that. You want to make sure that types is checked, so I do that. Make sure that CDS is checked, and then you want to make sure that the remaining fields are unchecked. So I'm going to uncheck all of these other options here. Then at the bottom, you want to make sure that tracks is checked, and then you should see a variance track, and make sure that that's checked. So this is what it should look like. This is just our sort of our default mode, by the way. Show annotations, types, CDS, tracks, and variants. And then if you look along the bottom panel, it'll say columns, track, and export table. So we want to go to the track option. Left mouse click that to highlight it. And there should be a variance track in here. We want to check that. Okay. And that's going to bring up another spreadsheet. And I've already configured this, so I have some columns already set here, but you should see some kind of a spreadsheet down at the bottom at this point. The next step is to go to the columns option, click on that, and it will bring up a fairly long list of column names or column headers that you can choose to configure the spreadsheet. You can see some of these are already checked, and I'll show you how we do that in just a second. But you can see there's a long list here. You have name, you have min and max genomic loci, you can have things like the length, the amino acid change, quality, CDS, codon change, and so on and so forth. All right, so you have all these different options. What you want to do is go to the top, click Manage Columns, and that'll bring up this view. And the list on the left shows all the available column headers that you can choose from. The list on the right shows the ones we've already selected. And what we can do, if you want, you can pick any available column, choose the right arrow, and that'll move it over to the selected column. So here it is over on the selected side. If we highlight that, if you want to rearrange the order of the spreadsheet headers, you can click the up button and the down button, move it around, whatever you want to do there. I've already got these in order. I don't really want quality in this report, so I'm going to move it back out to the available column with the left arrow. And now what I'm left with is the selected list shows all the column headers that I want in this spreadsheet. So then I click OK. And now what will happen is in the bottom panel, you'll see those headers show up here. So there's name, there's minimum, maximum, loci, the length of the polymorphism type, amino acid change, coding sequence, and so forth. And at this point, you can export the table. Just click on the export table option. That'll bring up a dialog box, and you can go to this look in field and choose wherever you want to store, whatever directory you want to store the file in. Down here, of course, we have the file name. There's a default naming convention. You can either leave that as it is or change it, whatever you like. You'll see that there's a couple of file types. One is the Excel CSV file. You can also use tab separated values or a TSV file if you want. We always just choose the default, just the standard CSV file, and then we save it. And that's really all there is to creating the variant spreadsheet file. Now at this point, we have the CSV file saved on our local workstation. Now there's something you can also format that in a different Excel format, so I'll show you how to do that right now. Okay, so I went ahead and closed out Geneious, and I launched Excel. I'm going to assume that you have Excel, some version of Excel installed. And one reason I want to walk through this is that most people want to convert the CSV file to an Excel formatted file that they can view and work with. And I'll show you how we do that here. It's pretty straightforward, but there's a couple of nice little tricks I can show you. Choose File, Open. That's all we're going to do. And then I'm going to look for the CSV file and just open that. All right. And it opens up the CSV file. Now the formatting's not great here, so we're going to reformat this a little bit. One thing I want to do first is we're going to save it in a different format. So we choose File, Save As, and it brings up this dialog box. And now again, this might be a little bit different on your system depending on what operating system you're using, what version of Excel, but somewhere it should say something like File Format. And by default it's CSV. What we're going to do is change that to XLS, sort of a standard Microsoft Excel format. And it'll say something like Microsoft Excel 5095 Workbook, XLS. The XLS suffix is what we're looking for basically. Click on that and then save it. So now the file is saved in an Excel format, XLS format. Okay, there's a couple of nice things you can do here. We can highlight the header record like this and go up and convert it to bold format. That helps a little bit. One thing I like to do here is go up to the View option. Hopefully you have something like this and freeze the top row. So now if we scroll up and down, the top row is frozen. All right. Then we can do something like I believe it's Edit Select All. We want to select the entire spreadsheet in this case, and then we can go up to Format, the columns, and there is this Auto Fit Selection option. And what Auto Fit does is it looks at all the cells and tries to reformat them so that every cell has sort of this optimal formatting. Just lays it out very nicely for you. Go up to Data and then Sort, because I want to sort this a little bit. Brings up this dialog box. The first thing I want to do is make sure I have the, it'll say something like My List has headers. Make sure that's checked because they do have headers. Go to the first field here and normally choose minimum, the minimum genomic loci. Click on that. And then we use smallest to largest in this case. Then you can go to the plus option, add another level, go back up here again. In this case, we're going to sort by the maximum genomic locus, okay, genomic loci, and then smallest to largest again. Click OK. And now what it's done is it's sorted this sort of across the genome from left to right, if you will, from smallest to largest genomic locus. And if we do a little scrolling here, you'll see that it's just kind of increasing across the genome. Okay, sort of left to right, smallest to largest. That's kind of nice because then we can just go scrolling through here and look at different polymorphism types. So you can see what kind of coding sequences there are, if there's any codon changes and protein effects and all that. And then, oh, don't forget to save that. So file save. And now you've got yourself a variant report.